This is the Elisa, and we will be wrapping up lab with this last test. And there's a short video that I want you to watch after watching this, and then you can do the lab quiz 3, which is so the Elisa utilizes antibodies. And we talked about this in lecture, how antibodies are made from B cells, and that B cells originate in the bone marrow from a stem cell there, a hematopoietic stem cell, that the B cells are made and they each have somewhere between 10 to the 14th and 10 to the 18th different kinds of IgD on the surface. So any given B cell has 100,000 copies of this IgD, a distinct IgD, and the B cell next door has a completely different set of IgD. Okay, another 100,000 copies, a totally different set, and the estimate is that there can be 10 to the 14th to 10 to the 18th different kinds of antibody made. Okay, once the B cells mature, and they mature in the bone marrow, that's why they're called B cells, they migrate to the lymphatic system to reside in the lymph node or the spleen. So this indicates here, this picture, that any B cell would have 100,000 copies on the surface, and we spoke of this in lecture, how we have to kind of think of the B cell as like a balloon, and on the surface, if you were to think of how many pieces of sand could fit, then this would give us an idea of the size that we're talking about, because proteins are quite small. Now these B cells, with their antibodies, they can detect antigen. And we said that antigen could be something like a spike on the surface of a virus. So this is what we see here, that this virus would bind to this antibody. So at maturation, B cells are taught to recognize self and non-self. Those that inadvertently react to self are deleted. That is called clonal deletion. But once a B cell that is now residing in the lymph or the spleen binds to an antigen, and in this case it seems to be a protein on the surface of a bacterium, the B cell that binds there would now begin to divide. It would get stimulated to divide. It would divide into two populations of cells, plasma cells that will make and secrete that particular antibody. It would be the same kind, that it would have the same affinity for that specific antigen. It would have the same variable region. We will see in lecture that it will have a slightly different FC region at the bottom, just slightly different, just how long it is. Not that it is an FC, it would still be there, it's just slightly different. So these would be IgDs on the surface of the B cell, but those that are secreted would first be IgM and then later IgG, which is what's shown in this picture. The other population of B cells, of course, would be the memory cells, and they would stay and they would remember in case this antigen came around again. We would mount an even faster response. Remember that a B cell is an antigen presenting cell, and so binding to an antigen would engulf that antigen. Yes, this microbe would be engulfed there, that portion, and now a portion of that antigen would be displayed on its surface, on its MHC class 2, and the T helper cell would bind there with its T cell receptor, and it would then stimulate the B cell with interleukins to begin this division to become plasma cells and memory cells. It would happen faster than if the B cell were to do it just on its own. So we said that there's different kinds of antibodies, And so that which is on the surface of the B cell is an IgD here. And you see it looks like our typical Y shape. That's on the B cell surface. So of course it would be found in the blood and in the lymph. It's kind of big. It doesn't have a very long half-life. It kind of goes away because it really wouldn't be loose. It would just be on the surface of a B cell. It has no known function in the serum. It's really just on the surface of the B cell. It's the B cell receptor. The activated B cell would begin to make and secrete an IgM, which is here the pentamer that we see. The IgM is great at agglutination because it can grab, 10 different things and drag them out of solution. So it was great at agglutination. And as I tried to discuss in lecture, the FC regions would all kind of be popped up such that they're very attractive to macrophage and to complement. So they do bind complement quite well. They don't last that long. They're enormous, so of course they can't get through the placenta, and they're very good against microbes and dragging them out of solution. This is our initial response. This is our initial antibody found in the blood. Presence of IgM usually means that we have an incipient infection. It's just started, because shortly thereafter, and this is where the T helper cells, cytokines come in, there's a class switch is what it's called, from an IgM to an IgG, and from here on out, that's all the plasma cell would make is an IgG response. And that makes up about 80% of any kind of antibody you would find in somebody's blood would be IgG. So it would be found in the blood, the lymph, and the intestines. A molecular weight of 150. This is our typical size. So you can see it's the smallest of all the antibodies shown on this table. It lasts 23 days. That's the half-life. So in 23 days, there's half as many. It does fix complement. It's the only one that can get through the placenta, and it does all the things that when we talked about what antibodies do, well, this is what we're talking about is primarily an IgG. So yes, it neutralizes toxin. It can neutralize spikes and adhesins so infections can't occur. It would also bind to complement. It would increase phagocytic response, and it would also be involved in the antibody-mediated attack of parasitic worms. That would happen as well if we encountered that. Okay, so I won't go into the others because that will be described in lecture, but this is just recognizing there's different kinds of immunoglobulins, different kind of. So they all have an FC region, which is typical to a species. So all humans have a very similar FC region, C for constant. And up here, they're made up of two chains, a heavy chain, which is the green one, it's long, and then the light chain, which is shorter. But up at the top of both the green and the blue one are the variable regions, and it's this that is different. It's here that the differences, the 10 to the 14th or 10 to the 18th differences would be here. So this is a lot of different amino acids in there, right? So when you think of 150,000 kilodaltons is what an antibody, let's say an IgG, is, any given amino acid is usually thought to be about 110. So you're looking at quite a few different kinds of antibodies there all together. So let's just do a little quick math here, 110. So we're looking at about 1,300 antibodies, 1,300 amino acids that make up an antibody. And yes, most of them are going to be in the FC region. You can see that's quite big, and even in maybe the constant regions of both the light and the heavy chain up here at the top. But the variable area is going to be, several hundred for sure. And when you think at each position you have 20 different possibilities, it does add up as how many different kinds of potential antibodies could be made. So we're going to do an ELISA. And if we did this in lab, then it actually doesn't take very long at all. And so it's an ELISA, which is a test that utilizes not one but two sets of antibodies. So an ELISA is a test to find an antigen, and ELISA is an acronym for enzyme-linked immunosorbent assay. So the immuno portion of the ELISA is because it uses antibodies to find antigen. Sorbent is because this test is going to take place, for us it would have been in a well, okay, a well, microtiter well, but for some other tests it'll take place kind of on a stick, okay, and I'll show some of that towards the end of this PowerPoint. So immunoantibody, sorbent, where the test is being done on a stick or on a well, okay. One of the antibodies has an enzyme linked to it, okay. An assay just means test. So it's a test that involves antibodies, enzymes, and a place for it to happen. Okay, so we have some absorbency going on. The antibodies, of course, are very specific, so we can use them to find a particular antigen. The specificity of the ELISA is because antibodies only bind to a specific antigen, and you know this. If you've had chickenpox, well, that's great. You won't get that again. You have antibodies and memory cells that remember that specific antigen on the surface of that particular virus, but it doesn't help you against pertussis. Okay, they're completely different, right? So pertussis is a gram-negative bacillus, and right, that's not a virus at all, and those antibodies are so particular to the chickenpox that they don't recognize this at all. So we can figure out if an antigen is around by using antibodies that would be specific to it. It's so sensitive because the ELISAs can detect tiny amounts of antigens down to 0.01 nanogram per milliliter. That's so tiny. Nanograms are 10 to the minus 9, and so this is 2 digits below that. Okay, so it's super sensitive, it's super specific, and we'll see that the specificity is enhanced not only because I'm using 2 antibodies, not 1, but also an enzyme. Enzymes are very specific too, aren't they? Okay, so how are the antibodies produced for this test? So for these ELISA tests, which are used all the time, an antigen that you're interested in. So let's just say right now we would really, and you maybe have heard people say this, what we need is the antibody test or the serology test, they will say, because antibodies are found in the serum, which is in the blood. We need that for the SARS-CoV-2 so that everybody can go back to work, right? So if you had the antigen, so let's say it's the surface protein of a virus, or it's a flagellin protein of a bacterium, something like this. An antigen of note, whatever one you want. Now remember what antigen means, antibody generator. You take that antigen and you inject it into a rabbit, 2 to 4 rabbits, just so you get enough of it made. That antigen will work on that rabbit just like the antigen would work on you. You would mount a response, it would be a specific response, and you would produce antibodies that would bind to that antigen. So after a couple weeks, then they take some blood from the rabbit, okay? And, you know, sometimes they give an additional boost of the antigen just to make sure that lots of antibody would be found in that blood. And that blood would then be screened, okay? They would first separate the serum from the blood, okay? And so now they would be able to remove the antibodies, and there'd be all kinds of antibodies in there because, you know, the rabbit has also been living maybe with an animal who's sick or whatever other antibodies that it's ever made in its life. You're not interested in all of those. You just want the one that binds to your antigen. So then it's poured over what's called a column. And so you see that the yellow triangles, that's our antibody. Excuse me, that's our antigen. So up here in the top left picture where you see the antigen, that's supposed to be here as well on the column. And now you pour the blood over this column that has the antigen, and only the antibodies that bind to the antigen will remain. So these are the yellow ones in this case. The blue ones would just wash away. You just rinse it off. Okay, and now this antibody can be removed from the antigen with another column that's not shown here, so that you would get a pure antibody to your antigen. Okay, that's perfect. That's going to be our primary antibody. That primary antibody, because it just came from a rabbit, okay, remember that the FC region of an animal is really specific to that animal. And so if you take that antibody, that primary antibody that I just made from a rabbit, and I couldn't find a picture that had a rabbit, so I'm sorry, just pretend this mouse is a rabbit. If you took that primary antibody and you put it into a goat, that goat thinks that antibody is an antigen. It does not recognize, of course it doesn't recognize the variable regions, definitely not. It doesn't recognize the FC either. So all kinds of antibodies would be produced from this antigen. Okay, in this case, the primary antibody is acting like an antigen. Okay, and that antibody that has been put into the goat is now forcing the goat to make its own antibodies against the primary antibody. This is called the secondary antibody. It too would be purified, okay, and instead of having antigen on the column as in the previous slide, it would have primary antibody on the column. You'd pour the goat's blood over, and only the antibodies that bind to the FC region of the primary antibody would remain. You can rinse it out, and now you have a secondary antibody. This secondary antibody, right here, this blue one, okay, which in our picture was made from a goat, is conjugated to an enzyme. In this case, horseradish peroxidase. This is very typical to use, especially in teaching labs. It's a very easy enzyme to work with. And so this secondary antibody is linked to an enzyme. That's the enzyme linked part. Enzyme linked to the secondary antibody. Okay, this is going to increase our specificity. So now we're ready to go. Okay, the primary will recognize the antigen, the secondary will recognize the primary, and the enzyme linked to that secondary will catalyze a reaction that will give us a color we can look at. So an ELISA is going to use both the primary and secondary antibodies, and that increases the specificity and the sensitivity, as does the use of the enzyme. So it's a very simple test. This is the well that we would be using in our lab. We would do it in triplicate, but I don't have a picture of that. You can take somebody's sample. Okay, it could be from their saliva if I was looking for if they had mono. I could take their blood if I was looking for HIV. Okay, and put that sample into a well. All kinds of stuff would stick on the surface of that well. Antigens are primarily proteins. Proteins have amino acids. Some of those are charged, and these wells are plastic. That's partially charged, so there's like a charge-charge interaction going on here. You let it sit for a while, then you wash it out. Okay, a lot of stuff just gets washed away, but some antigens would remain. Now you add your primary antibody, which binds to the antigen. Okay, you leave that in there for a while, wash off any excess antibody. And now you add your secondary antibody, which binds specifically to the Fc region of the primary antibody because it's from a different species. So if our first antibody is from a rabbit and our second is from a goat, that rabbit antibody doesn't look right to a goat. That's why it produced the secondary antibody to the Fc region. That secondary antibody has the enzyme linked to it. That's the letter E here. Okay, that's all rinsed away, and that's rinsed 3 times to confirm that I don't have any loose secondary in there with an enzyme on there, because now I'm about to add the substrate, and if the enzyme is there, it will react with the substrate as enzymes do, and it will give me a color that I can measure. And it's really easy, a very visual test. You don't need to have any science background to do in ELISA, because it's really taking somebody's sample, urine, blood, could be stool, and going through a series of adding solutions, primary, wash steps, secondary, wash steps, and then a substrate, and boom, you've got a color. It's an instant yes. Yes, if there's a color, that means that the secondary antibody is there, and if that's there, it's because the primary is there, and the only reason the primary would be there is that the antigen is there. So yes, Mrs. Smith, you have mono. Okay, just ELISAs are used all the time. You maybe have even taken one. If you've ever used a pregnancy test, this is what it's looking for. Not at this time, it's not looking for a virus. This is not something for illness, but a pregnancy test is looking for human chorionic gonadotropin, HCG. Human chorionic gonadotropin is something that is made by the placenta, okay, initially in the first stages of pregnancy. Notice that you have a certain level of it at, well, week 0, you're of course at 0, but once you get pregnant, the amounts raise, okay, until you're about 12 to 14 weeks in, and then it starts to kind of tail off, okay? So you have some human chorionic gonadotropin that is being released in your urine, and so that works by having this pregnancy stick. You kind of pee on the end of the stick. That's how this works, and it's dry, and so then the urine wicks up. So it wicks up first, and if the human chorionic gonadotropin, which is going to be our antigen in this example, it will bind to the primary antibodies which are there, and these have the enzyme on it in this example. It will wick further, and here we have antibodies that recognize human chorionic gonadotropin also. And so now notice it's sort of like this is called a sandwich ELISA, because the antigen is in the center of 2 antibodies. This would produce 1 line. If the human chorionic gonadotropin, if the HCG is there, we would get this line, because this secondary antibody that you see at the bottom is specific for HCG. However, there's a second antibody that just recognizes the Fc region. This is drawn a little bit weird, but it recognizes the Fc region of that first antibody, and the antibody will bind there. Now, if you just get the second line, that just means, okay, the test is working. That works as a control. Yes, there is antibody in here. Yes, wicking will move it forward in the stick, and it will bind here to this antibody, which recognizes the Fc region. But if you get a second line here, okay, That's the test line. The only reason you could get that to bind is because this first antibody here binds to the human chorionic gonadotropin. Okay, it's not binding to the antibody with the ball on it, the antibody with the enzyme. It's not binding to that one. Okay, so you might, if you're pregnant, you should get 2 lines, the control line plus, yes, you have human chorionic gonadotropin. Okay, that's the second line. And if you're not pregnant, then you just get the single test line, the control line. Okay, so that's, not an illness, all right, but I can use enzymes and antibodies to test for a very common human chorionic gonadotropin hormone that would be present in pregnancy. Okay, so the swab is used to take a sample from somebody's throat, and then now the swab is inserted into this area on the stick, and it's a little bit wet in there, so we have the same wicking action. Okay, and so notice that we have a control line, okay, to indicate that yes, the antibodies are moving, that there are antibodies in there, that the antibodies do react. Okay, that's very important. In any test, you've got to have a control. So the antibodies would move and indicate that yes, this test is working properly. Okay. If you get the first line, that's the test line, that means that yes, you've got strep throat, that the antigens present on the Streptococcus pyogenes, the causative agent of strep throat, are present, that they have bound to that first antibody, okay, which then binds in this T line, binds to a second antibody, which is also specific for the antigens present on Streptococcus pyogenes. Okay, so if you have 2 lines, it's because yes, you have strep throat. 1 line means no. So that's a very typical thing and super easy, and they can do it right in the doctor's office very, very easily. It doesn't take very long at all. You can test for norovirus in the exact same way, except that norovirus is a virus that is fecal oral, and so you're testing for stool. So ELISAs are used all the time. They're super easy, and now that you are getting the idea behind an ELISA, maybe you're understanding the push currently to get an ELISA, an antibody test for the SARS-CoV-2, because it's very easy to use. It gives you rapid tests. It has high specificity for that antigen. It has great sensitivity. It can recognize antigens at very tiny levels. And so this would be something that, almost anybody could do this. You wouldn't need necessarily to go to the doctor's office to get this test. Certainly if you came up positive, you're going to have to find some health professionals to help you get over this disorder, etc. But at least you could get a yes or a no. So I'm certain they are working on this and just getting it to be really specific and getting the antibodies made and antibodies purified. I went over it in 2 or 3 slides. Like, it's no big deal. It's a big deal. But once it's done and made, then it works great and we're all happy. So that's pretty much the end of this ELISA, and you'd be ready now to take, after watching the video, then you'd be ready to take the quiz.